So the last time we talked about the developing brain and the influences which can change the fate of the developing brain and thereby the fate of the young fetus, the embryo, and the young neonate. What you see happening and what you saw happening was that a normal growing brain, if malnourished, if deprived of appropriate hormones, or if incorrect hormones are added, or if stress is given, so many things can happen and can change the brain forever, irreversibly, which means that a normal growing brain can become a damaged brain. So we were talking about the possibilities, the enormous chances of things going wrong if we don't look after the mother and if we don't look after the growing fetus. Let's go on into the other influences. There are many more. We talked about nutrition, we talked about hormones, we talked about what happens if we change different kinds of hormones. Now let's go a little further into the oxygen levels. One extremely important area, and we know, you know, every living organism requires oxygen to survive. Similarly, for the fetus and the embryo. Now, this question was raised a little earlier when we were discussing the development of the young. Where does the fetus get its oxygen from? Yes, you've thought about it. I'm sure you know. Take a guess. From the mother, like other substances, it comes from the mother. You and I breathe. We take oxygen from around us. Now, where does this fetus get? It's floating around in the embryonic fluid. Get its oxygen. Take a guess again. Yes, through the mother's blood circulation. And which particular component of the blood circulation would that be? Of course, the red blood cells. So that brings us to what happens if the mother is severely anemic. If the mother is severely anemic, and I've said this many times, that the fetus is at the mercy of the mother, because whatever it gets is from the mother. And if the mother is deficient in oxygen, so will be the fetus. And if the mother's blood circulation, because this is what brings oxygen into the fetal system, and this is what cleans the fetal system, also the circulatory system. And what if there is deficiencies in these systems? So, if the mother is not getting enough oxygen during the last trimester, what happens? Hypoxia of the fetus. Hypoxia means lowering of the levels or absence of oxygen, the required amount of oxygen that is needed. The mother's blood circulation, if it is faulty, will damage the brain areas which may have developed normally. So, you have a normal brain, you have a normal grown brain of the embryo, and just by not providing the right amount of oxygen through the mother, the respiration, cells die off. And again, this is an irreversible damage. So, damage to the brain can take place when mother's circulation is faulty or mothers may not be providing enough oxygen through anemia or heart disorders or other problems which have a faulty circulatory system. This damage is similar to the damage of mental retardation. And this also happens in birth trauma. When the baby is born, immediately after that, oxygen is not provided or does not have the capacity to take in oxygen. The mother's oxygen ends with her system. So the newborn is now on its own mercy. Hypoxia during birth trauma has also led to damage of the brain. Why? Why is oxygen so important for the brain cells? Just like it is important for you, me, and every living organism to survive. So, when the cortical areas atrophy or die off, lesser area remains functional. And which is why, if less oxygen is available, cells will die off, and when cells will die off, lesser brain remains functional, lesser number of cells remain functional. And the later that this happens during the embryonic ages, the more the damage is permanent. During the early stages, there is possibility of reversing the damage, but not later. So, remember, oxygen is extremely important. Then, other influences, drugs. The fetus, again I emphasize, is at the mercy of the mother. Now, being at the mercy of the mother means whatever the mother takes, whatever is the intake of the mother, is going to be passed on through the placental barrier into the system of the fetus. There was this very important case that I told you earlier of thalidomide, and we talked about the thalidomide babies. Thalidomide is a bromide, a sedative which was given to mothers during the early trimester, the first trimester, and this was to counter morning sickness, and this was done in the 1960s. The mothers felt a little better after taking this thalidomide, but when their babies were born, they were extremely deformed, and this deformation led to an awareness that whatever substances mothers take, and this was the extreme form which we have seen, that whatever substances mothers take, the infants are at they also, whatever stage they were at the point in development, that particular organ was affected or malformed. Physically, these babies were malformed. And there was a huge case on that pharmaceutical company which had to pay damages. But that was a permanent damage. You could not grow limbs back. You could not grow lives back, right? So, that was what began a look into the influences of drugs which the mothers take and how it influences the development of the fetus. My research was on opiates, and while I was doing research, and I would suggest you read this, whether it is morphine, heroin, cocaine, marijuana, you know, these drugs with young boys and girls take, and then they assume nothing will happen, but it sits in your system, and then once they are addicted, they cannot get off these substances, especially women, when they are expecting, even nicotine. So, all these substances are passed on to an otherwise normal, healthy fetus. And there was one paper in which there were reports, of course, when I was doing my study on the effect of mother's addiction on the neonate or the newborn. While the neonate is born, let me take you back to the fetus. While the fetus is in the system, the fetus gets its supply of drugs from the mother. The mother takes morphine and it's passed on, so the fetus gets addicted too. What a horrifying thought, right? Mother takes cocaine, passed on to the fetus. The malformations, whatever they are, will be taken, but the basic is that fetus becomes addicted. So, what happens when the fetus is born, neonate? The mother's supply of drugs end with birth. Umbilical cord gone, born, independent. And what happens is that this terrible, terrible, terrible pain that this neonate goes through, because it is going through withdrawal, no longer gets the drugs from the mother. The mother supply has ended. So what happens? And there were cases that were reported of intense, extremely excruciating, painful withdrawal which the babies, the newborn babies were going through. In fact, these babies have to be given sedatives to control that withdrawal. How painful? So, mothers taking drugs, heroin, alcohol, cocaine, nicotine, severe deformities apart from the withdrawal do occur. And therefore, now you will see the label on cigarette packs that Surgeon General has warned that expectant mothers should not, etc., etc. So, any substance that you take or a mother takes should be very careful, because it is going to have a irreversible effect on. So, there is a responsibility of the mother. So, mothers are warned against taking any substance which they can do without, and which might affect the development of. So, pharmaceutical companies have to have these labels, warning, and there, when we do pharmacology, and we'll come to that point, you'll see that there are substances which have teratogenic effects. And teratogenic effects means they have effects on the growing fetus cells. So, neurodevelopmental and teratogenic effects, where they have done these researches and found out that mothers should not be taking these drugs if they were expecting. And there is a certain period that is also very clearly given. So, drugs, very dangerous, and we have to be extremely careful, because by just not controlling ourselves, of course, that sometimes it is beyond the control of the mother, but then you seek help. Drugs can irreversibly damage the brain and the body of the growing fetus. Then we have the prenatal motor activity. One of the things that we talked about earlier. Is there prenatal motor activity? Yes, it is. There is. Around the 4th month, you see the baby, the fetus is moving around, the embryo is moving around. So, there is a lot of activity and exercise going on inside. So, increased activity in the uterus leads to stronger muscles. So the more the embryo is active, the fetus is active, the embryo is active, the neonate is active, has a stronger muscle. Now, why would that happen? Remember, go back to the studies of Hubel and Wiesel, go back to the studies where brain connections are being formed, and external stimulation we talked about becomes important when the target organs are being reached out by the neurons. So, that is the time when we know that exercise, the sensory connections and motor connections have to be strengthened, and external stimulation becomes important, but the template and the plan is laid out during the exercise in utero. Recently, in the last few years, mothers have started singing lullabies to their young fetuses, embryos. And research has shown that these, when born, these as neonates, these babies are responsive to their mother's voices. So they have been primed during the in utero life to the mother's voice. So there is external stimulation and there is some prenatal motor activity and some prenatal sensitization which takes place, which is good. Remember the birds sitting on eggs while hatching? The chicks do recall and do remember and do sing and do respond to the way their mothers did, chirp. So there it is, similarities between the birds and humans. External stimulation in another form, and we had talked about the sensory deprivation. We had seen experiments of Hubel and Wiesel, where when the eye was taken out, deprived, the connections in the brain were not formed. So we must realize that this has happened in humans as well, and very interesting experiments that have come to light and very interesting cases have come to light. And these were the cases of the wolf children. You've heard of the wolf children, I'm sure? No? Okay. You've heard of Romulus and Remus? No? You've heard of Rome? Yeah, you heard of Rome. Rome was founded by two twins, Romulus and Remus. And the legend goes that these two were wolf children. And who are these wolf children? Are they now also found? Yes, wolf children are children brought up by or in a pack of wolves. Does this really happen? Yes, it does. And I have seen some films, actual documentaries taken of young boys, girls who were brought up with the wolves, and when they were separated from the wolves, what do you think? They started walking like us? I mean, if they were brought up with the wolves and the wolf cubs were their friends from early postnatal period to about 3-4 years of age, 5 years more, 11, 12, they walked on all fours like the wolves. They barked like the wolves. They ate like the wolves. They snarled like the wolves. Even though they had the capacity to talk, they could not talk. Even though they had the capacity to stand up and walk, they could not walk. Why? Among other things, remember, it is the external stimulation coming from the environment. When a child learns to walk and is taught how to walk, that is external stimulation. The mechanism is there. So when the external stimulation is missing, there is an entirely starved system where the centers in the brain for all these activities, they atrophy, and no amount of training will bring that center back. The template is changed, and whatever stimulation is given, it grows according to that stimulation. Okay, then the complex environment, which means, of course, that you need to stimulate a growing neonate. How do you stimulate a growing neonate? We stimulate babies. How do we do that? There are many ways. Of course, mothers talk to babies. That you have seen. And what else? People take babies from one hand to another, from the grandfather, grandmother, cousins, brothers, everybody will hold them and different handling will take place. Then, when baby grows a little older, you walk with the baby. You teach them how to say words, right? And you're very happy when they copy to you. They say, mama, so you look at that, oh, it's a word. And that is external stimulation. But even before that, now you see cots, cribs, where there are little mobile toys and different shapes and different, so the baby keeps looking up at different colors, different shapes, different sounds. And the more the external stimulation, the more stimulation would be the connections. Remember, the connections are being formed in the early period of life. So that is very important. And now comes a point where very stimulated mothers read stories to their growing neonate. And it is said that they do attend, they do respond. Interesting. But the more you stimulate, the more rewards in terms of growth of the brain and sensitization of the brain to more stimuli. This brings us to another area, which is, what if we have a complex environment, providing a complex environment to the growing neonate? Rosenweg, late 60s, early 70s, provided very classic findings. And this you can see in most psychology books, where there is a rat cage in which rats have fun things, wheels and toys and colored balls and swings to play with. And in another, there is a simple cage, a steel cage, just the water and the food. Which brain do you think is more enriched? The brain which has a complex environment, where the rat has to go through the wheels, through the swings, through the balls to get to its food or to get to its home. So, the young rat pups learn the complex environment, and thereby there is a sprouting of more dendritic connections, therefore a more intelligent and efficient brain. So, complex environments are good for the development of the brain. What does this also show? The evidence from Rosenweg study. There was, just like in nutrition, there was increased brain weight, larger cerebral cortex. So, a heavier brain. What does that heavier brain mean? Of course, you have already, because you know that the nourishment will bring in more cells, more cerebral thickness of the cerebral cortex. Thicker cerebral cortex means more cells. Thicker cerebral cortex also means more connections, more fibers. So, thicker cerebral cortex, larger number of neurons, larger number of dendritic sprouting, larger number of connections, and larger the size of neurons as well, more glial cells, and of course, the enzymatic activity, which is chemical composition of the brain cells, is different as well. These cells are active. So, you see, just by providing a complex environment which the young neonate has to manipulate through, the brain is growing better, faster, bigger, more efficient. Greenup in 1975 also showed that there is increased dendritic sprouting, and also the rats that are in a complex environment, bred in a complex environment, respond in the same manner as the mildly handled rats. Do you remember who are the mildly handled rats? Levine's study, 1960s. Mild handling was good, was beneficial, mild stress was beneficial. So, here you had these rats which were stimulated, and the rat brains which therefore benefited from this complex environment. Goes to tell us that yes, you need to have a complex or a stimulating environment for the brains of the young babies. And of course, living in a culture like Pakistan, where everybody is involved in the lives of the new babies, in most families, this is what happens. It is good. Also, and if your grandmother always tells you, कि बच्चे को जी हिलाएं जुलाएं, इसको पड़ा ना रहने दें, है ना, मालिश करें उसकी. सब कहते हैं कि जी वो अंग्रेज़ों की किताबों में लिखा हुआ नहीं, ऐसा नहीं करना. दादी कहती है नहीं, इसको तेल लो ना, मालिश करो इसको नहलाने से पहले. क्या है ये? Yes, that is stimulation. That is sensory stimulation. That is physical stimulation. And that is what gives the brain more stimulation to grow and to remain. Of course, it strengthens all the connections, and apart from that, it provides a connection, an emotional connection between the mother and the young. So, it's good. Some of the things that, maybe most, but unless we learn it from the West, we will not say that it was good, right? So, आज जाके दादी माँ को कहें आपको बहुत कुछ पता है, और मैंने मेरी जो साइंस की किताब है, साइकोलॉजी की किताब है, और न्यूरोलॉजिकल बेसिस ऑफ बिहेवियर वालों ने भी यही कहा. शी वाज राइट. Okay, so external stimulation and the stimulating environment is extremely important. But again, remember, it is only during the periods that the brain needs to grow, or the period then it is that particular boundary in which it is needed. Neither before nor after would that make a difference. So, you see, we have completed, and I have walked you through this particular topic, where you started off from the germinal layers and then right up to the neonate, how the brain has grown and how the brain is being influenced by so many factors, so many miracles that happen in every juncture of the development of the brain. And what I always say is, it's always a miracle that a baby is born healthy, because as you see, there are so many things that can go wrong. So, be careful. Be careful. Even coke is a drug. Even aspirins are drugs. And even some substances that you take in a food. So, be very careful. Radiation, X-rays, बगैर सोचे समझे हम जाके एक्सरे कराते हैं. That also, हिरोशिमा की deformities अब तक पता है. So, and deformities of the long 3 Mile Island, Chernobyl, many massive accidents of radiation, people are still going through that. Then poisons or toxins like mercury, they also influence the development of the brain. So, be very careful of what you eat, what you drink, and where you are, what you are breathing. Lead, that makes our future more vulnerable. So, do read final, do read up on material that you can get your hand on in developmental psychology, developmental neurobiology, and ponder about it, think about it, because this course is meant to make you understand and also make you see how science has brought realities to us. We have finished this topic. We will now move on into the next topic, which is topic number 5, which is neuroanatomy. So, after you have known about the brain and how it grows, now we are going to go into the sections of the brain, open it up and see what is inside the brain. This is adventure of the highest order, right? You are excited about it. So, let us go and look inside the brain and see what we find there. So, now we are going to be talking about basic neuroanatomy, the topic number 5, in which we are going to be talking about the systems, the structures, the cells, the nervous system, especially the central nervous system. Peripheral nervous system is good, but the action is in the mastermind, which is the central nervous system. So, keep your notes handy and keep your notebooks with you, and we are going to walk you through the brain. What an adventure. When we talk about the brain and the spinal cord, the brain and the spinal cord form the nervous system, and the central nervous system, the components of the central nervous system have similarities. The brain and the spinal cord have these similarities. They have neurons and they have glial cells. The basic building block of the nervous system, the basic brick on which the foundations of the nervous system lie, are the neuron and the glial cells. And of course, cerebral spinal fluid, and we are going to talk about that as well, but mainly let us start with the neurons. What are these neurons? Neurons are very highly, and they are like other cells in the body. They have the similar structures and similar life cycles, and they have emerged, if you remember, from the same germinal cells, but that is where similarities end. Neurons are specialized cells of the nervous system to receive, to integrate, to assimilate, to transmit, to conduct information using electrical and chemical means, and many things in between. So, neurons are highly complex factories which are buzzing away with activity. I move my finger, I move my hands, I move my tongue, I speak. The sounds that you hear, hundreds and millions of neurons are buzzing away so that I can talk. And what I am thinking before I talk. So, neurons, extremely important cells with functions which are similar but very different from the rest of the body. These are chemical powerhouses which have a life of their own and a thinking of their own. And we work through that. There are about 10 to 12 billion neurons in the brain, and there are many different types of neurons, and we will see some of them as we go along. There are 4 major types that we can talk about. One is the unipolar neuron. The unipolar neuron is one string or one fiber, one going from point A to point B, and this has only, it is like this, one particular cell body attached to this fiber, unipolar, one pole, only one. The bipolar is that neuron which has, as the name implies, 2 poles, bi polar. One is the receiving end and one is the output end. And the receiving end is again a single line, but there are two different kinds of fibers. The receiving end is the dendrite, and the output end, which comes out of the cell body, is the axon. So the bipolar is the unipolar, and the bipolar are simpler forms of cells. Bipolar is found usually in your sensory system. Remember, if you have seen the structure of the retina has in its receptor cells the bipolar cells. So the ganglionic, the bipolar, and then the rods and cones. Bipolar, more effective, more efficient for immediate communication, passing on information from one end of the pole to another, one end of the neuron to the other. So it's quick information coming in, being processed, and quick information going out. Then comes the third category of neurons, which is the category which is found mainly in the brain, that is the multipolar. The multipolar, as the name implies, has many extensions. Only one axon, every neuron has only one axon. So there is, for all neurons, there is only one axon. It may have, axon may connect to axons, but one axon, which is the output end, and it is a neuron which will have more processes going out of it, and not just more processes out of it, but it will also have the branching of these processes. So there will be dendrites, and these dendrites will have short branches, and these branches will then branch out further. Very intricate set of network that this multipolar, assume that this is a multipolar neuron. A neuron which has a body, one axon, and multiple branches of dendrites going out of it, and multiple dendrites branching and connecting and passing messages into these, and then again branching out. Extremely intricate system. And these are also of different kinds. Golgi type 2, Golgi neurons, which are found in the cerebral cortex. Those which are shaped like a star, bunches with short axon and short dendrites. Why would you think they would be like this? Because quick information. And then there would be the spindle cells, where they are going across different layers of neurons of cerebral cortex. So there are many different kinds of multipolar cells. There are interneurons also, which have short or sometimes no axons, and these are for quick integration of or processing of information. My favorite multipolar neuron is the pyramidal, shaped like a pyramid. And there are pyramidal neurons found within the cortex, different layers of cortex, and the pyramidal neurons have one dendrite which is very long, several millimeters long, going from one layer of the cerebral cortex right into your leg or some other extremity. So it's a one long dendrite. Axon may be long, may be short, but the dendrite is very long, and it is called the apical dendrite. So there are many different forms of multipolar neurons. Neurons are clustered into nuclei, or into bunches, and these bunches work together, aggregate together. Remember aggregation? They are together because they have similar function and they will perform similar function. So they are connected to each other. They form these, think about like a bunch of grapes, right? So they're bunched together. And just like the grapes, individual grapes are tied to that main branch, in the same manner, there are fiber tracks which connect these nuclei. So we have nuclei, neurons in bunches of nuclei. We have fiber systems which are connecting these nuclei, and we have then another close neighbor, the glial cells, which are, if the glial cells were not there, neurons would not be able to perform the kind of functions that they perform. Neurons are there in place and maintaining their form and their structure and their filaments and their dendritic spines. I always ask my students, how come, this is extremely fine, fibrous filaments, these dendrites and the branches of dendrites. How come they are always in place? They are always connected to each other. How come when you jump up and down, they don't go jiggle, 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 jiggle? Have you thought about that? Otherwise, the whole brain will become a jelly. No, it doesn't. Because within these, there is the glial cell. Now, where do you think the name glial come from? It came from glue. Makes sense, right? So the cells are almost glued together. Neurons are glued together by the glial cells. So they maintain their positions, they maintain their connections, and they maintain their insulation. So glial cells are supportive cells, and these cells hold the neuronal systems together. And these cells also do what we call the housekeeping jobs. Now, what are the housekeeping jobs? घर में क्या होता है? क्या चीज़ें आपको घर में हाउसकीपिंग लगती हैं? खाना पकाना, खाना खिलाना, आपको घर की सफाई करना, चीज़ें जब आप स्कूल को वो यूनिवर्सिटी को भागे और अम्मी मेरा बस्ता कहाँ, अम्मी मैंने फ़लाँ किताब रखी किधर है, वो पकड़ाई जानी, राइट? So the housekeeping jobs, among others, is keeping the place clean, which is removing all the debris from the there are the intracellular spaces and the extracellular spaces. अब इसको थोड़ा सा एक्सप्लेन करती हूँ मैं. इंट्रासेल्युलर, जिन्होंने बायोलॉजी नहीं पढ़ी उनको लगेगा ये एक्स्ट्रा सेल्युलर और इंट्रासेल्युलर स्पेस क्या होती है. एक ग्लास पानी का सामने रखें और उसके अंदर एक कतरा ऑयल का फेंकें. अब क्या नज़र आएगा? वो तो अपनी अपनी जगह पानी में ऑयल का कतरा फ्लोट हो रहा. बिल्कुल इसी तरह से आपका जो एक्स्ट्रा सेल्युलर स्पेस है उसमें सेल जो है वो फ्लोट कर रहा है. एक्सट्रीमली जो बड़ी फ्लूइड लाइक सिचुएशन है. अब इस फ्लूइड लाइक सिचुएशन में उस सेल को अपनी शेप, अपनी फॉर्म, अपने कनेक्शंस मेंटेन करने के लिए चाहिए है ग्लियल सेल. So glial cells provide the support, provide the connections which are maintained. यहाँ supportive cells हैं और ये मैं आगे जाके आपको बताऊंगी कैसे. They also keep the place clean. जो extracellular space है उसमें वो सफाई करते रहते हैं. ये वो है janitorial service. So they move out the dead cells. अब आप पूछेंगे डेड सेल कहाँ से आते हैं? भाई इतने सारे न्यूट्रिएंट्स आ रहे हैं, इतने सारे सेल्स कंटिन्यूसली और बाकी फाइबर्स वो जो चीज़ चाहिए नहीं है वो निकाल के सेल बाहर फेंक देता है, उसको किसने सफाई करनी है? इन्होंने. The glial cells move the dead cells out of the nervous system. Otherwise, if the dead cells keep accumulating, you know what will happen? Brain will get toxic and die off. Cells will become dangerously filled with necrotic material, dead cell material. So, they need to move this dead cell out of the system so that it is excreted and the brain kept clean. It also provides nutrients. Cell को कहाँ से मिलते हैं? The glial cells will provide those nutrients because cell ने खुद तो नहीं बाहर जाना, but cell is a living powerhouse generating energy and working constantly. And we'll look at those pictures and you'll be surprised as how a busy system the neuron is and how much activity is happening within that micro system. इसमें बहुत सारे किस्म के ग्लियल सेल्स हैं. एक का नाम आपको पहले भी मैंने जिक्र किया था, एस्ट्रोसाइट्स. एस्ट्रोसाइट्स, as the name implies, they look like stars because they have these branches extending out of them. And these astrocytes are used for a particular purpose, and or they have a purpose within the brain, and we'll talk about that. Then there are oligodendrocytes, and then there are phagocytes and microglia. For every neuron, there are about 10 to 12 glial cells. So, अगर 10 billion neurons हैं, तो 10 raised to the power of 10 to 12 glial cells. कितने ज़्यादा? So, it is an extremely crowded brain that you have up here, especially considering the fact that their extensions, their fiber systems, their synapses is also there. Yeah, बहुत ही buzzing, this little thing up here is buzzing with activity, and all these neurons and these glial cells are buzzing away with activity and energy. So, what do we have in this? If you look closely, the glial cells are, as I said, classified into 3 main groups. The astrocytes, I want to cover the glial cells and finish them before I move on to the neurons. Not that glial cells are not interesting, but there is much more to talk about of neurons than of glial cells. So, coming back to glial cells are of 3 major kinds. The astrocytes, which are the largest glial cells found in the brain, and they are, of course, as we said, star shaped. Then we have the oligodendrocytes, and the oligodendrocytes, बड़ा मुश्किल नाम है oligodendrocytes, they are basically made up of fatty lipid sheets and multiple sheets, and they form the myelin. ये बहुत दफ़ा नाम आपने सुना है, myelin covering, right? तो myelin covering form करते हैं oligodendrocytes. And then there are microglia, and then there are phagocytes. Microglia and phagocytes are also glial cells, but of a very small structure and a very small size. Astrocytes. Astrocytes क्या हैं? Astrocytes basically are glial cells which perform a very important function. And this important function is that all the blood capillaries and the blood vessels which enter the brain. Now, the brain has to have nutrition, has to have oxygen, has to have substances brought into it. And where does it come from? Right? It comes from the circulatory system. Yes. What an effective system it is. So, through the blood vessels and through the capillaries, we receive the material within the neurons and within the glial cells and within the brain itself, which is needed by it. But you do recall all the stuff that you eat every day, not all is good for the brain, right? But it does enter your brain supply. So what happens? How does the brain protect itself? कभी सोचा नहीं ना? It does protect itself. And who protects the brain? The astrocytes. Around the blood vessels and around the capillaries, the thinnest capillaries. Capillaries होते क्या है? बहुत 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 thin blood vessels. ये वो blood vessels हैं जो कि आपके smallest area, micro areas के अंदर तक पहुंच जाती हैं to provide the blood. So, what happens here? That these astrocytes cover, insulate and protect the blood vessels. Now, this forms what is extremely important for us. Remember this. It is known as the blood-brain barrier. So, astrocytes, by covering the blood vessels and the blood capillaries, form the blood-brain barrier. Any substance that has to go into the blood vessels or come out of the blood vessels into the brain has to go through these tightly knit astrocytes. So, ये molecules के movement जो है इन astrocytes के बीच में से गुजर के, that is what protects your brain. So, at all neuronal cell bodies and their branches, जहाँ जहाँ neurons हैं और जहाँ जहाँ blood vessels के साथ interaction होता है, वहाँ वहाँ astrocytes उसको insulate करें. They also provide nutrients and chemicals which pass through the blood into the cell. इसको protect भी करते हैं और they also act as a medium through which or a bridge through which these nutrients get into the brain. So we have talked about, कैसा लगा? Interesting है ना? The getting into the brain and understanding how the brain, you've just started. I would suggest you do some reading. कुछ नहीं तो तस्वीरें देख के देखें. तस्वीरों से ही आपको बड़ा interesting लगेगा. और अगली दफ़ा मैं I'll walk you through some pictures of the neurons, the glial cells and of course the cell body, and you will see that we are working in a very intricate, हमारा काम आसान होता है because neurons का काम intricate है. Do some reading, do some thinking, keep your eyes and your ears open and do regular reading. Otherwise, this is a technical field. पहले हम biology में थे, फिर हम psychology में चले गए, अब हम chemistry में जाएंगे और biochemistry में जाएंगे. So this is what neurological basis of behavior is. Do read your handouts, do your homework and do check out some websites on neurosciences. See you next time.